Human immunity. Physical and chemical barriers are used as the first line of defense to stop infection from entering the body. The body's initial line of defense against pathogens entering the body is the innate or non-specific immune system. It's also referred to as the non-specific immune system because it reacts the same way to all pathogens and foreign chemicals. Parts of the first line of defense include skin and mucous membranes, as well as tears. The skin is an example of a natural barrier to infection. The closed surface means that germs cannot enter. The skin tissue also contains specialized immune cells that aid in the defense against invaders. The skin is home to a variety of communities of helpful microbacteria known as the skin microbiota. Tears are another part of the first line of defense against pathogens. Tears contain an enzyme that breaks down the cell wall of most bacteria. Tears wash out foreign objects that enter the eye, and additionally, tears include a substance called lysosome that has an antibacterial effect and acts to stop microbial invasion and infection. The second line of defense is the immune system, so the group of cells, tissues, and organs that work together to protect the body. It responds very fast, for example, ensuring that germs that have entered the skin through a minor wound are found and eliminated within a few hours of their entry. However, the innate immune system has not very much ability to prevent the spread of infection. Inflammation is part of the second line of defense. So inflammation's primary purpose is to create a physical barrier that prevents infection from spreading. Additionally, it removes dead cells and tissues harmed by the first assault, and the inflammatory process starts tissue repair and eliminates the primary cause of cell injury. Blood flow increases during inflammation, aiding in the delivery of immune cells to the infected area. An infected area close to the body's surface turns red as a result of this increased blood flow. White blood cells and fluid are able to enter the damaged tissue through the blood vessel walls, which become permeable. There are three main roles of macrophages in inflammation. These are antigen presentation, phagocytosis, and immunomodulation through the production of different cytokines and growth factors. Macrophages are essential to the beginning, continuation, and end of the inflammation process. Fever is also a part of the second line of defense, as it is a rise in body temperature which is intended to suppress bacterial development and speed up the immune cell response. Macrophages are a type of white blood cell that are crucial to the functioning of the human immune system. They are also part of the second line of defense. They perform many tasks, some of which include engulfing and digesting pathogens, removing waste products and dead cells, boosting other immune system cells. M1 and M2 macrophages are the two basic types. So the M1 type, which is known as the classically activated type, are triggered by pathogen invasion and have a significant impact on immune response to diseases like bacteria. The M2 type, known as alternatively activated, they are anti-inflammatory and aid in tissue regeneration and wound healing. Non-specific defences, or innate immunity, describes aspects of the first and second lines of defence. Innate immunity, which is also known as non-specific immunity, is the defence mechanism you were born with. You are shielded from all antigens because of it. Barriers that prevent hazardous substances from entering your body are part of this innate immunity. So, non-specific defences, or innate immunity, include aspects of the first line of defense, such as skin, mucous membranes, and secretions of the skin and mucous membranes, but they also include aspects of the second line of defense, such as antimicrobial proteins, inflammatory response, and fever. And then specific defenses, so adaptive immunity, include aspects of the third line of defense, which include lymphocytes, antibodies, and memory cells. The third line of defense is acquired specific immunity. The third line of defense seeks to eradicate particular infections that the immune system has already encountered through adaptive or acquired immune response. The adaptive immune response spreads throughout the body, so not only to the infection site. B lymphocytes, otherwise known as B cells, and T lymphocytes, otherwise known as T cells, are two primary types of white blood cells that make up the adaptive immune system. T cells are involved in cell-mediated immune responses, whereas B cells are involved in antibody-mediated immune responses. B cells are activated in antibody-mediated immunity when they come into contact with a known antigen. The antigen is subsequently engulfed and digested by activated B cells. Antigen MHC interaction further activates T helper cells, which release cytokines that cause antigen-presenting B cells to develop into antibody-producing B cells, otherwise known as plasma cells. Some B cells are now converted into memory cells to maintain the immune system's readiness for future pathogen invasion. The largest type of white blood cell, monocytes, are produced in bone marrow and account for between 5 and 10% of the total amount of white blood cells in a human's body. Eosinophils are crucial cells in terms of allergies and worm infestations. They make up between 0.5 to 1% of the white blood cells in the human's body, although this number may be larger if you're currently experiencing allergic reactions. Neutrophils are the most prevalent form of white blood cell in the human body. Between 40 to 60% of white blood cells in the human body are neutrophils. They have an uneven nuclei and are medium-sized. The organisms are consumed by neutrophils through a process called phagocytosis. Lymphocytes are tiny spherical cells with a large nucleus. They're produced in the bone marrow, after which the lymphatic system contains them. Between 18 and 42% of the white blood cells in your body are made up of these, and B or T cells can develop from lymphocytes. And finally, the least common type of white blood cell is a basophil, which can be found in a variety of human tissues. They release histamines to assist in preventing allergic reactions, and anticoagulants, which are blood thinners and antibodies. Immunoglobulins. Antibodies, also referred to as immunoglobulins, are glycoprotein molecules made by plasma cells, which are white blood cells. They play a crucial role in the immune response by selectively identifying and attaching to specific antigens, like a virus or bacteria, and assisting in their eradication. So an antibody is a large Y-shaped protein that the immune system employs to recognize and destroy foreign substances, like dangerous germs and viruses. Antigens, which are distinctive molecules of the infection, are recognized by the antibody. Sensitivity to antigens. Antigens are specific proteins on plasma membranes of different cells or pathogens. When they are phagocytosed by macrophages or other APCs, the antigen can be presented to lymphocytes. There are major compatibility complexes on the APCs presenting the antigens to different randomly made B lymphocytes. Then if one of them fits via clonal selection, they will proliferate, which is clonal expansion. This creates a bunch of cells specific to that antigen that go and produce antibodies against it. Some of these cells differentiate into memory cells and circle around in case of that same pathogen invading the body again. Self-recognition and tolerance. The body's capacity to discern between self and non-self is known as self-tolerance. Through the use of antigens, the immune system accomplishes this. So major histocompatibility complexes, MHC, 1 and 2, and human lysosomal are two groups of cell surface proteins that the immune system uses to distinguish between itself and foreign substances. Therefore, if an MHC cell is present on the surface of an antigen, the immune system will not attack or will indicate an attack against it because it's recognized as self. However, the adaptive immune system will react if the immune system encounters a known antigen, that has not yet been seen by it. The immunological response to from the human body to exogenous antigens is not constant, so the degree of response varies greatly depending on how much the new antigen resembles the self. The act of transplanting organs, tissues, or cells from one person or another in order to replace a damaged organ with a healthy organ is known as transplantation. However, the immune system has developed potent defenses against foreign invaders and serves as a powerful barrier to the transplantation process. Self-markers designate a cell as a component of the self and are present on the surface of every nucleated cell in our body. Each type of organism has unique self-markers. The immune system of the recipient will detect that the new organ is distinct from the body's other tissues and will launch an attack in the form of rejection. Rejection can be avoided, however, if the organs are closely matched. The organ is as closely matched as possible thanks to tissue type, but no two people have the same antigens unless they are identical twins. The autoimmune disease lupus occurs when the body's immune system assaults its own tissues and organs. Lupus-related inflammation can impact a variety of bodily functions, including joints, skin, kidneys, blood cells, brain, and heart. Because of how frequently its signs and symptoms resemble those of other illnesses, lupus can be challenging to identify and diagnose. Some people are predisposed to lupus from birth, which can be brought on by illnesses, medications, or even sunshine. Lupus has no known cure, however, medicines can help manage the symptoms. Rheumatoid arthritis is a type of type 3 hypersensitivity. It's a chronic autoimmune condition that mostly affects joints. Warm, swelling, and uncomfortable joints are frequent symptoms, including flare-ups when symptoms get worse. When someone has one autoimmune disorder, like lupus or rheumatoid arthritis, it's quite easy to develop one or another because they frequently do co-occur. Although the specific cause of high prevalence of co-occurring autoimmune illnesses is unknown, we do know that many autoimmune disorders don't only attack one portion of the body. Diagnosing autoimmune disorders requires identifying the antibodies that the patient is producing. This is done through blood tests and is much more accurate than relying solely on symptoms to diagnose. Unfortunately, there's no known cure for autoimmune illnesses, thus methods of treatment typically focus on managing the symptoms. Acquired immunity is immunity that someone develops during their life, so they're not born with it. Active immunity is gained from exposure, so either natural exposure to infection or vaccinations, which is artificial exposure. Passive immunity is acquired from someone or something else, like antibodies transmitted through chest-feeding babies, which is natural, or antibodies acquired from serum medicine, which is artificial. Active immunity is immunity you develop, whereas passive immunity you require from somebody else. Herd immunity, vaccination programs, and factors affecting vaccine uptake. Herd immunity is the concept that those who do not receive a disease vaccination may be protected to some extent if a sufficient amount of the population has received protection. Herd immunity can be attained by vaccination programs since they help expand the population of vaccinated individuals. The widespread use of vaccines, or the proportion of people who have them, depends on a variety of factors. This includes cost, access to healthcare, and public awareness. To ensure that the population is appropriately protected against disease, it's crucial to address these variables. The body's acquired immunity is mostly made up of herd immunity, vaccination programs, and factors affecting vaccine uptake. Herd immunity helps protect those who are not immunized, and vaccination programs and other factors that affect vaccination uptake help to enhance the overall vaccination rate. Vaccinations are capable of protecting the body from illness and infection. To evaluate the benefits of vaccination, it's crucial to look at measles infection rates in the UK between 1940 and the present. Before the vaccine was developed in 1968, measles infections were common in the UK. However, since the introduction of the immunisation, measles infection rates have steadily declined. According to the World Health Organization, measles mortality decreased in the UK by 99.7% between 1940 and 2017. This highlights the value of vaccination in general, as well as the efficiency of the measles vaccine. The data on successful vaccination in the case of measles in the UK is a clear example of how well vaccination protects the body from infection and sickness. Reasons for reduction in vaccine uptake. There are many reasons why vaccine uptake may fall. Donald Trump was US president at the beginning of the COVID-19 epidemic, and he was on the record as having anti-vaccination attitudes. Due to his incredibly large influence, Republican voters were significantly more concerned about vaccine safety than Democrats were. There also may be a reduction in vaccine uptake even when people do want protection from the virus. Not everybody has available transport to a doctor's surgery, and others may avoid medical professionals due to bad associations or past trauma. Many doctors' offices only have working hours during weekdays, and poorer people may not be able to take time off work in order to protect their health. People from certain minority ethnic backgrounds may have hesitation to receive the COVID-19 vaccine, for example. This has been linked to a lack of faith in both government and healthcare providers, brought on by institutionalized racism and prejudice. Immune response to vaccination. An essential component of acquired immunity is the immunological response to vaccination. Antigens are injected into the body during vaccination to elicit an immunological response. Different immune cells, such as B and T cells, which create antibodies known as immunoglobulins, are involved in this response. These antibodies aid in recognition and elimination of foreign substances like bacteria and viruses. Immunological memory, which is the body's capacity to remember an antigen and react to it more quickly in the future, is developed by the body. This immunological memory is a crucial component of the body's acquired immunity and aids the immune system, and this is strengthened by vaccinations. The immune system is strengthened by vaccinations, which helps to prevent sickness and infection. Immunological memory. Some offspring of B and T cell replication develop into long-lived memory cells. As a result, if the disease ever infiltrates the body again, the memory cells might trigger a powerful reaction since they remember every unique virus the animal encountered during its existence. Antibodies provided by the mother throughout fetal development and through breast milk are what confer passive immunity. The infant is protected by the short-term memory until its own adaptive immune system is fully developed. Memory B and T cells help to quickly wipe out the pathogen during a secondary immune response, preventing reinfection by the same virus. For a pathogen that has been weakened so it cannot cause infection, the antigen of a pathogen is delivered to the body during vaccination. Without really producing the symptoms that the virus causes, this encourages the immune system to establish a specific immunity against that pathogen. Due to frequent strain alterations and difficulties in creating an immunization potent enough to work but not potent enough to spread an infection, there aren't vaccines available for every pathogen that exists.